Thanks for joining me. My name is Dario Skefos, and I'm with the MetaCell team. We worked during the Codeathon to develop a tool to integrate two different pieces of software so that you can visualize neuronal networks from SCKAN in the Open Physiology viewer. And so what does that mean? SCKAN is kind of the pathway at the bottom here where you're taking models that have started as NLP models, and then you've got expert curators that are working to refine those neural network models and put them into a database. But then Apinatomy is a methodology for expert curators to create a circuit diagram showing all of the different connections throughout anatomy so they can represent physiology in an anatomical context. And we thought it would be really cool if you could take these models, all of the models coming from this Spark connectivity knowledge base, and represent them in Apinatomy so you could learn a lot more and explore how they work. So this shows the tool SCKAN Composer that we've previously been working on, and we have ongoing projects to develop. And then this makes very simple point A to point B network diagrams, and you can actually expand those over time as we continue to develop the tool. Apinatomy, on the other hand, has been going on for quite a while and has a lot of different complexity that you can add in there. So you can add multiple scales of anatomy coming from organs down to tissues and cells and subcellular locations. And the same schematic can be used across all sorts of different species and across all sorts of different physiological scenarios. So the approach for this project was to take the latest release of the database coming from SCKAN, and that comes out as a turtle file that then needed to be converted to a JSON and in turn converted to a spreadsheet, which is what the Open Physiology viewer can use. So it's a little more friendly for people to create new models. And the asterisks on this diagram show the different components that we worked on for this project. So basically, all of the models that were given to us are able to be imported into the Open Physiology viewer. And I'll show you a quick little video of what that looks like. These are some of the models being brought in, and you can also see that you're able to see it in the context of the whole homunculus of all the different organs. It shows kind of spleen and thymus and things like that. So I'm just going to proceed. This shows also the extension of a model to show a comparison between sexually dimorphic development, because a lot of the models that were given were about the reproductive system. So we showed the female versus the male in pink and blue. And then we provided links to some additional models that were provided. So this is our team. Please reach out with any questions, and we look forward to continue to work on these projects. Have a great day.